These are the directions for the PyMol Lab Assignment. The first thing that we need to do in this assignment is to search Google for the Protein Data Bank, or PDB. Click on the first link that comes up in Google, and this brings us to the PDB homepage. What we can do now is to search the PDB for a particular protein structure. The structure that we're interested in looking at today is 1MBO, which is the PDB code for a specific myoglobin structure. Here's the entry page for myoglobin. We can see here that there's a picture of the structure itself, information about the publication, but the one thing that I'd like everyone to look at in the beginning is this ligand chemical component information. This is a nice thing to look at in the beginning before you view a structure, because it gives you some idea of what to expect from the non-protein components of the structure. For example, here we know that there's going to be a heme in the structure. We know that there's going to be molecular oxygen, which is going to be represented as this oxy in the structure, and there's also a sulfate ion. This is a good thing to know before we look at the structure so that we know what we're looking at when we start viewing it. Let's go back to the top and download the file itself. Click on PDB File Text. This is going to download the correct kind of file to be viewed in PyMol. That file is going to go directly to your Downloads folder, and then go ahead and retrieve it and then open the file in PyMol. We're now looking at the structure of myoglobin in the PyMol program. Let's start by expanding the PyMol window so that we can get a better view of what we're looking at. What we see is a single myoglobin protein in green sticks surrounded by hundreds of red water molecules which have been crystallized along with the protein. Let's start by practicing some of the mouse commands in PyMol. The left mouse button can be used to tumble the structure. Click and hold somewhere on the structure and move the mouse. Click and hold again, move the mouse. Do this over and over again. This allows us to rotate the structure and allows us to see the structure from any side. Let's use the middle mouse button now. The middle mouse button is usually a scroll wheel. Don't spin the wheel, just click and hold the scroll wheel and then move the mouse around. This allows us to pan the structure. This can sometimes be useful if we need to change our angle of view. Next, use the right mouse key in order to zoom. To do this, we're going to click and hold on the right mouse key and then push the mouse up and down. This allows us to zoom in and out, giving us different sizes, different views of the structure. You can use all three of the mouse keys in conjunction in order to get any kind of view you want from your structure. I'll also mention some other mouse functions that can be useful for us. You can use the left mouse button a different way. You can bring the cursor over to the right or left part of the screen, click and hold, and move up and down. This allows us to spin the structure. This is different from the tumbling. The tumbling happens when we click on the structure and move the mouse around. The spinning happens when we go to the edge of the screen. So this can be really important if we need to just spin the structure to get a different view. Another important function of the mouse is to change our angle of view. So now use the mouse scroll key to actually scroll. You can see some things that are disappearing now. This allows us to change the angle of view and eliminate certain information. This can help us if we want to highlight certain parts of the molecule by eliminating something in the foreground or in the background. One problem that sometimes crops up in PyMol is the problem of a lost structure. What I mean by that is that sometimes the pivot point of our image becomes too far from the molecule itself, and the molecule seems to pop in and out of view, and it becomes very difficult to control what's going on with the structure. If this ever happens to you and you lose your structure, simply come up to the top right and click on the Orient button. This will now reorient your pivot point so that you can control your structure again. You can also solve this problem of a misplaced pivot point by using the middle mouse button or your scroll wheel. Simply come over to the structure and click once using your scroll wheel. This will also reorient your structure and restore a more easy to deal with pivot point. Now that we understand how to use the mouse, we're going to start using some of the other functions of PyMol. The first thing I'd like to do is look at the sequence of the myoglobin protein. To do that, come up here to Display and click Sequence On. What that does is it gives us this long string of green letters. This is the primary structure of the protein. You can see the first amino acid in the crystal structure is valine, followed by leucine. Those are residues number 1 and 2. If we use the scroll bar by clicking and holding on the scroll bar, we can actually view the entire primary structure of the myoglobin. We can also see some of those ligands that we noticed in the PDB entry page from before. This is the sulfate ion. This is the heme. After the heme, we see this long string of what looks like zeros. These are actually the oxygens from the water molecules which surround the myoglobin. These are also numbered. Let's click and hold on the scroll bar and see all the waters which are in the crystal structure as well. And at the end, we see this oxy. That oxy is the oxygen molecule which is being coordinated to the heme in the myoglobin. All right, let's scroll back to the beginning of the amino acid sequence, and we're going to start modifying the structure. We're going to start modifying our view. Let's go down to the selecting state. This is a really important feature of PyMol to know about. What we can do with the selecting state is we can toggle through different kinds of selections. We're going to learn all about what these different selections do, but let's first toggle over to objects. So to get to the objects, just left click on selecting state until you get to the pink objects here. What I'm going to do now is I'm going to come up here to the amino acid sequence. You can click anywhere on the sequence. Don't click on the numbers though. Make sure you click on the actual letters of the amino acid sequence. Left click once. And what that does is it's selected the entire object. That's why we changed the toggle. We toggled into selecting state objects because now we've selected the entire object. Every atom of the object is now selected. Okay, we can click again to turn it off, or click one more time to turn it back on. So we've selected everything. What I want to do right now is we're going to right click and we are going to hide everything. We're going to hide everything. And then click somewhere off in the black part here, and what we've done is we've actually erased our entire structure. Now the reason we erased everything is so that we can build the structure up again. Come down here to the selecting state and toggle over to molecules. What we're going to do now is we're going to scroll over to the beginning of the structure and then just left click once on the amino acid sequence to select the myoglobin structure itself, right click, and then show cartoon. Left click on the black part to deselect. And now what we have is the secondary structure of myoglobin shown in this cartoon format. We can see here it has several alpha helices and some unstructured loops, all in green, and this is giving us some idea of the secondary structure of the protein. Something important to notice is that all those red stars, which represent the water molecules, have now all been eliminated. This is because we selected the entire object, got rid of it, and then selected only the myoglobin molecule. All the water molecules were not included in that selection. So now we have a much clearer view of what we're looking at. That's probably a very important thing to do when you're looking at most structures, to eliminate those water molecules because they really distract from what we're trying to understand about the structure. Let's just scroll over back to those water molecules again, and let's go ahead and highlight some of them. We're still on selecting state molecules, so now we can actually select each water molecule individually. I'm going to select and hold and select several of them, and then I'm going to right click and I'm going to show these guys as spheres. All right, so those are the water molecules that we had just erased. They're not particularly interesting or important for what we're trying to do right now, so we're going to go ahead and get rid of them again. Select all the water molecules by left clicking and holding, right click, and then hide everything to get rid of them again. Always remember to unselect when you're done dealing with a certain kind of selection. Next, we're going to show some of the more important features of the myoglobin. Obviously, the secondary structure, the overall picture of the molecule is very important, but the ligands that are bound to the myoglobin are actually very, very important, so I'd like to show those as well. Let's go down to that selecting state and toggle through until we get to residues. Residues is going to allow us to select individual amino acid residues one at a time. The residues that I'd like to highlight in this structure are histidine 64 and histidine 93. let's use the scroll bar to find those residues. Histidine 64 is H64, which is right here. That's the histidine at position 64. It's not the 64th histidine. There aren't that many histidines in the structure. It's the histidine at that position. We're also going to scroll over and find histidine 93. Let's find histidine 93 right here. And then you can see that they've been kind of selected, even though they're still invisible in the structure here. Hover over one of those selections, either this one or this one, doesn't matter which one. Hover over it, and then right-click, Show Sticks. This is going to render those histidines in the stick model. So we can use our left mouse button to rotate the structure a bit, and let's zoom in a bit. And then we can see those nice imidazole rings of the histidines. All right, these histidines are going to be really important because they are part of the coordination environment for the heme and also for the oxygen, which is bound to the myoglobin. Let's use the scroll bar to find those other ligands. Let's go find the heme. Left click on the heme. They treat, even though the heme is not an amino acid residue, it's still treated as a residue in PyMol. So left click on that heme ligand, then right click, show sticks, same thing as before, and now our heme has appeared. We can now see why those histidines are important. Those histidines are, one of them is coordinating the iron, which is this orange atom in the middle, and the other histidine is going to be important in coordinating the oxygen. Let's go find that oxygen. So use the scroll bar to come over to the very end until we find the oxy. Left click on the oxy and then show sticks. So it's a little hard to see, but there's our oxygen, our O2 right there. So this structure is looking very nice. It's a much clearer rendering from what we started with those lines, those green lines and the red stars for the water. We're starting to really get a picture of what the myoglobin looks like and where its important ligands are. The next thing I'd like to do is show you some of the other features of PyMol that allows us to change and customize what we're looking at. Let's go ahead and go back to the selecting state and toggle over until we find molecules. What I'd like to do now is change the color scheme for this entire molecule. Right now the molecule is using green carbons. If you look at the imidazole group, you can see that the carbons are in green and the nitrogens are in blue. This is okay, but I think a more standard color scheme is going to use a gray colored carbon. So let's go ahead, and we're on selecting state molecules, so we're going to select the entire myoglobin molecule. Left click somewhere on the on the primary structure to select everything. Right click and go to color by element, and then come down here and select the one that had the gray carbon. Do you see how we have these different colored carbons with different color schemes? Let's go ahead and click this guy. So now the molecule has changed color to the gray carbon color scheme. The nitrogens are still blue, the oxygens are still red, but we've gotten rid of that kind of green color. You can also play around with other color schemes. Another interesting color scheme is to color it by a spectrum. And what this does is it allows us to see the N and C termini of the molecule very easily. The N terminus is always going to be blue, and the C terminus is always going to be red. So this is nice to see the directionality of the polypeptide chain. But for now, what we'd like to do is just the standard color scheme. Go back down here to the gray carbon color scheme. Now that we've changed the color scheme of the molecule, we can go ahead and customize it even further. What I'd like to do is render some of the atoms of the molecule as spheres. This will allow us to highlight certain parts of the structure even further. So let's go in and zoom into the heme. What we can do now is directly click on the heme itself and also on the oxygen molecule. One thing to notice is that you can click to highlight things in the molecule view screen or in the primary structure screen. So you can click up here on the heme to highlight and de-highlight, or you can click right on the structure itself. By the way, aiming your mouse is very important. If you're a little off, like if you're on the black part here, it won't really work. So you have to really click on the exact spot that you're trying to highlight. All right, now that we've highlighted the oxygen and the heme, let's go ahead and right click to show some of the atoms as spheres. So here the aim is very important. Put the mouse right over one of the atoms and right click. If you don't have it over one of the atoms, it may not work exactly right. You're going to get this other screen, which is not what you want. Okay, what you want is the screen that shows the different selection options. Okay, so aim with the mouse directly over one of the atoms. That's the right click, and you're going to click on show, and then spheres, which is right here. Left click on spheres, and now some of our atoms have become spheres. This gives us a bit of a more realistic view, maybe of the correct size of this heme and the oxygen. What you can do now is left click on the black part to deselect and take a look at what you have there. Now you have a heme and the oxygen, kind of more of a blown up and more realistic view of what it could look like. So although this view of the heme is a bit more realistic in that it gives us a better idea of the size of each of the atoms, it's very difficult for us to read and for us to understand what that structure really is. So what we're going to do is we're going to customize the size of those spheres so that we can see it in a more normal way. Let's go up to the Setting menu and then click on Edit All. Edit All gives us a window with several hundred parameters that can be changed. These are all the different parameters that can be changed in PyMol. Use the scroll bar to scroll down to sphere scale. Sphere scale, which is going to be right here, sphere scale. Left click on sphere scale to highlight it, and then double click it. All right, so double click it so that we can change the number in this value screen right here. So 1 is the full size of the sphere. What I want you to do is click on the 1 and then change it to 0.3 and then click enter. So what that did, it reduced the size of the spheres by over 1/3 and it shrunk it down. Click on done to exit out of this screen. Be sure not to click on other buttons, including the reset button, which shows up on the PC version. You don't want to reset the values because you'll just change everything back to what it was originally. Click done. And now we can see what we've done. We've changed our heme so that it's in a ball and stick model, kind of like an organic chemistry model kit set. And now we can really nicely see what's going on here with the oxygen, which is coordinated to that iron atom in the middle of the heme. All right, we're almost done rendering our structure. Let's zoom out, use the right mouse button key to zoom out a bit, and take a look at our myoglobin structure. You can use all your mouse button keys to explore the structure. Really the main function of PyMol is to explore the structure. So using all your mouse buttons to get all the different angles is really, really important. The last thing that I'd like to do is to show the surface of the myoglobin molecule. This gives us an idea of the overall topology of the structure. To do that, we're going to toggle down here, back to selecting state. We're going to toggle over to molecules because we want to look at the surface of the entire molecule. Again, click anywhere on the primary structure of the myoglobin. You can also click somewhere on a helix to highlight the whole thing. That's another way to do it. So click on the entire myoglobin molecule and then right click, and then we're going to show the surface. And then left click on the black part to deselect. And what that does is it's giving us an idea of the overall topology of the molecule, maybe something close to the true shape of the molecule. One thing that you can notice right away is this really cool hydrophobic pocket where the heme and the oxygen are kind of nestled in there. And one interesting thing to notice is that these carboxylic acid groups from the heme are sticking out into the solvent, while the hydrophobic parts of the heme are pointed into the center of that hydrophobic pocket of the myoglobin. So this structure is okay, but I'm going to customize it a bit more. One thing I find a bit distracting are all these different colors. The reason there's these different colors here is it's showing you which atoms are solvent exposed. So every time you see the blue patch, that means that there's a nitrogen on the surface there. Every time you see the red patch, it means that there's an oxygen there. It's not very important for getting a basic idea of the structure, so I'd like to change that and make it a little bit less distracting. So again, we're going to go up to this Edit All window, and we're going to customize the view of the surface of the molecule. So to do that, we're going to change the surface color. So to do that, we're going to scroll down until we get to surface color, which is right here. We're going to double click on that. Right now it's on default, which means that it's going to be whatever color is the atom is underneath. We're going to change default. We're going to click white and click enter. And what that does is it changes the color of the surface to white. Click done, and now we have sort of a much more simple view of the structure. Maybe that's easy to see what's going on. One thing that's missing, though, is the view of the inside of the structure. So we've kind of obscured some of the information by showing the surface. What we can do now is actually make the surface a bit transparent so we can see both the structure on the outside and the secondary structure, all those helices inside. So let's go up to, again, we're going to select the entire structure, and we're going to go to Setting, Transparency, Surface, and we're going to change the percentage of transparency for the surface. Click on 40%, left click to deselect, and now what we have is a view of the inside and the outside of the structure at once, so we can see some of the underlying architectures as well as the surface topology itself. At this point, we've completed editing our PDB session file. Let's go to the top and save it so that it can be opened later in PyMol. Save the file in a place where you know to find it later. And now we're going to create the actual image that will show up in our Word document for this assignment. The first thing that we need to do is to change the background color to white. This will allow the structure to show up a lot better in the Word document. The next thing that we want to do is consider how we're going to frame the image. Do we want to show the whole molecule? What angle do we want to show important parts of the molecule? What I'd like to do for this structure is to angle the structure in a certain way that shows the important side chains that we highlighted. Also to show the O2 molecule and to show some of the hemes so that we can get an idea of the complete structure of the important part of the molecule. And let's also zoom up. Don't be afraid to zoom up and fill the frame with the important part of the structure that we're interested in. Now that we've decided on a framing for our image, it's time to create a high-resolution two-dimensional picture that can be inserted into a Word document. To do that, we're going to go up here to the top right and click on this button, Ray. This is going to ray trace the image. Depending on the computer that you have, it might take a few seconds for the computer to complete the calculations. An important thing to be aware of is that if you were to left-click anywhere on the structure right now, you would actually undo the ray tracing and you'd have to re-ray trace the image. But for now, we have our nice ray traced two-dimensional image that can be saved as an image file. Okay, this is different from the session file, which is an editable three-dimensional file. The PNG file is an image file. It's just a normal two-dimensional image file that can be inserted into our Word document. Let's save this also. And now we've actually completed the PyMol component of this assignment. To finish the assignment, we need to insert the new image file into a Word or a Pages document. To do that, we'll open Pages or Word and insert the photo from the picture file, and then we find the PNG file that we just created and insert our image. We'll do the same thing in the Pages program. We're going to go to Insert, Choose, and then find our picture that we created, the PNG image. And there we've completed the assignment. Notice that I've included a nice figure caption that gives a very good explanation of what the image is showing, including some of the details that we're showing, especially the residues that we highlighted. Alright, so we've completed the assignment. Save the file as either a PDF or a Word document and submit it to Turnitin.